This is the first of a series of videos that we're going to be doing. It started off as an idea for basically grad students that had never seen search algorithms before. And it blossomed into this idea of kind of based off of a baking show where you come in and you have a developer actually walk themselves or me through a search. So from start to finish, how do you get software, data, everything? And then really, I think more importantly, it was an excuse for me, Ben Neely, to get to talk to people that I respect a lot and haven't gotten to meet or do know and just want to hang out. Our first entry into this is the good folks at MetaMorpheus, and I'm going to let you do your introductions.
I'm Rachel Miller. I am a fourth-year graduate student in Lloyd Smith's group at UW-Madison, and I'm part of the MetaMorpheus development team, and happy to be here talking about what we do today.
Yeah, so I'm Rob. I'm a graduate student. I've been in Lloyd's group 4.5, something like that, years now. My main project's Flash LFQ, which is the quantitative part that MetaMorpheus uses, which we'll talk about later. But I've also been sort of lead maintainer on MetaMorpheus for about 3 years now, and that's transitioning to Rachel because I'm hopefully graduating soon. So she's now officially sort of lead maintainer, and I'm sort of lead maintainer emeritus.
Hi everyone, my name is Mike Shorey. I've been in Lloyd Smith's lab for well over 20 years. Like I don't even know the count after that. And doing all manner of analytical chemistry, but for the last sort of 10 years or so, I've been involved in proteomics, and MetaMorpheus actually got sort of started on my watch back in the day, and it came out of an early collaboration we had with Josh Kuhn's lab, in particular a student named Craig Wenger. Craig Wenger created a simple program called Morpheus that was used to search high resolution tandem aspect data, and it was like super fast, and it was like really simple to use, and it worked really well. And we had some ideas about doing some PTM discovery work, and we didn't know anything about programming or software or whatever, but he was on campus here with us, and so we struck up a collaboration, and you know, out of that eventually grew MetaMorpheus, which is sort of like solely under control of the Smith lab now, and we named it MetaMorpheus in honor of Craig's early work. So that's kind of a shout out to him.
I mean, that's great. That was my first question, by the way. We were gonna start there. So and not to beat you to the punch, but one of the points of the show was I have a colleague who is always convinced that software doesn't actually run. And so what I'm really excited to see here is like we go, right, so it's a fresh install, nothing's configured as far as the software goes. We're just vanilla, click the buttons.
Yeah, we're gonna try, and you know, if it like totally, you know, dies, I expect we'll like insert video of like some big explosion or like whatever, but you know, we're gonna attempt. So yeah, go ahead and like chime with any questions, but I'm gonna take over the screen now, and I'm gonna go to Google, and you know, we're just gonna try it. Here is my search window, and I'm gonna type in GitHub MetaMorpheus. And so all of our software, you know, MetaMorpheus is just one of the programs that we write, is available on GitHub. It's totally open source. We use a very liberal MIT license, so people can kind of use it however they want. They can repackage it, repurpose it. We want anybody that wants to take advantage of the work that we do to have access to it. And so everything's here on the GitHub. You have to scroll down a little bit, okay, to finally sort of get to the text here. There's a couple things that I sort of like want to point out. Right here where it says download the current version here, we're going to click that just in 1 second, and then that's going to sort of download it to my machine. But before I get there, I just want to talk about some system requirements, okay, so it'll run. All right. This is the kind of key one. If you're running in a Windows or Mac or Linux environment, you need to have compatibility with .NET Core 3.1. Okay, so it will work in all of those environments. There's no GUI for Mac. but you can run it from the command line in Linux, provided that you have the .NET Core 3.1 installed. I have a bit of a bigger machine, but we recommend at least 8 gigabytes of RAM, and it's naturally multi-threaded. So the more cores you have, the faster it's going to run. Here we go. And so by clicking that button, it's going to automatically take you to the most recent release. Once you have it installed, every time there's a new release, it will notify you when you open MetaMorpheus up. Okay. For Windows applications, we're going to download the installer here at the top. But if you want it to run in the Linux command line, you would download this command line.zip and just operate it from there. Rob's going to talk about that a little bit later, how to run the command line and so forth. I'm just going to sort of stick with the GUI for now. So here we are, we're going to save the file, and I am opening up my downloads, and you can see here's the installer right here at the top, and I'm just going to double click that. And while I'm waiting, I'm just going to, oh, it says don't run, but really you should. You have to click on more info and run anyway. Next, install. All right, and we're just going to launch MetaMorpheus. And there you go. So MetaMorpheus is now totally installed.
Now, you had said that every time that you kind of do this in front of an audience, it's somewhat surprising.
I'm completely shocked. Every time that works without that. You know what I mean? I don't know. I shouldn't be at this point because it's been around a long time. But I had the same experience your friend has, and I have had forever, which is that nothing anyone else ever wrote ever works really fast. And I from early days, I wanted this to be so easy, even I say this, so easy even a PI could do it. And so so that's kind of been our goal in terms of making it sort of user friendly. Before I'm gonna show you one more thing on the GitHub before we launch into this. Okay, we're gonna go back to the main MetaMorpheus page, and this is really handy for new users. Okay, right down here we have an explanation of how to do some test data and how to do how to test your installation is working. Okay, so if you open up this box folder here, what you're going to see is there's a couple folders. One folder's got some MZML files that have already been converted from .raw. One folder's got FASTA database files, and then there's a number of search tasks here. Okay. In addition to that, we have the 2 original raw files. These are from mouse data that we published a while ago, and I think after we save this video, we can provide links to that down in the description. And then there's a couple additional style databases. Rachel, in just a minute, is going to explain about how to use tasks and so forth. But I just wanted to show all of this stuff is here, and I already have it downloaded on my machine. And so that's what I'm going to use to start this video out. But I just wanted to explain how we got the data that we're using for this test. So this is the MetaMorpheus vignette file. You'll recognize that those are the same files that we had before. Okay, so here's the GUI, and the GUI is arranged kind of how you use it in order. All right, and so the first thing you're going to do is add in the databases. Then the next thing would be to add Spectra. The third thing would be tasks. In this case, the task is going to be a search task. And then the last little thing is run. All right. As I said, Rachel's kind of go through this in greater detail in just a second, but I'll show you how this works. So if you click on databases, you can see that there's an empty area where files go. And I can just simply drag one of the FASTA files from here down here, and it will just drop in. We have already curated a list of contaminants. And if you want to add default contaminants, you can just click that button. Okay, so now you've got a contaminants file. If you decide later you don't want to use that, you can uncheck the box, or you can just hit the delete button and it'll go away. But we'll just throw it on there for now. Okay.
Can you just, if I imagine someone What's in your contaminants file? Is this like about 100 things?
Exactly.
Or-
Like a whole variety of things. Originally, we started with the CRAP database. That was the beginning. And we've adjusted it a little bit since then. Dr. Brian Fry in our lab poured over it quite a bit. And it also, we've added PTMs to those things. And so Rachel talked about using XMLs, but most CRAP contaminant databases just have the base protein amino acid sequence. Ours also includes PTMs from those organisms. And when you get the results, it'll show you whether it's a target protein or a contaminant protein. Okay, so you have the option to filter, because you can imagine there's like human keratin in here. That's a common contaminant. But if you're using a human database, okay, by default, it won't use the contaminant human, right? It'll sort of exclude things from the same organism. You get the choice to exclude those from the same organism. Okay.
So an example of like a PTM on a contaminant is like methylated trypsin, because like trypsin manufacturers methylate their trypsin so it won't auto digest. So like our contaminants database will help you look for those things if you want.
Wow. No, that's
also has like some of the components of like cell culture media that can get carried along. So bovine serum albumin, etc., to look for in case you don't fully wash everything off of your cells before lysis.
Yeah, that's I mean, I love to hear about the complexity of that, because for those people, when we say CRAP, that's the common repository of advantageous proteins, right? And that was kind of an old one from, I guess, Ron Beavis, right?
Yeah.
And that had things like, even had like latex and wool, but you're like kind of taking it up a notch, which is things I didn't know. So that's really cool.
I mean, I think people like to use it, but nobody ever really knows what's in there. And I think some people kind of use it sort of blindly. To be honest, ours started with the CRAP. We also borrowed heavily Max Quant, Andromeda search engine. They provide a contaminants database. We also like looked at theirs. And so we, combined some of that. So, like full disclosure, standing on the shoulders of giants here, this isn't like a product of our own work. It's it's a incremental improvement on some excellent scholarship from other people before us. Next thing is Spectra. You see the same basic panel. Okay, so I'll go back up here to the vignette, and I'm going to just grab raw. This is a thermo raw file format. You can also use .mzml. I'm going to drag this in here, and you're going to see at some point soon when I try to run this, it's going to prompt us to like agree to a thermo license. Okay, but it will read thermo raw file data natively, so you don't have to do any file conversion if you don't want to. Like, you can just sort of save the time.
So, not to interrupt. So it's MZML, MGF, or RAWs.
That's correct.
Okay. So, but like with other tools or writers, you could make those, right?
That's right. If we have a second at the end, I think Rob is going to just pop MS convert up quickly and show you the parameters that we use to convert from like raw to the MZML style that we like. I do want to point out that we do not favor MGF, and this may be getting into the weeds a little bit, but we like to deconvolute the spectra and pull out additional IDs, and oftentimes MGF kind of throws away MS1 spectra and it just gives you whatever precursor mass. That's like a much diminished data set. We would really much rather have MZML or .raw because we can pull a lot more IDs out of it. But you can read MGF. All right, and so we're on to the tasks here. And you see there's a number of tasks: calibration, PTM discovery, search, crosslink search, and glycosearch. Right now, I'm only going to talk about search. Okay, so you add the search task, and what it's going to pop open is it's going to pop open the parameter tables here. We hid a lot of stuff down here at advanced options. I'm not going to talk about any of this stuff because I want people to sort of see. I'm just going to add the task, and that's all you need to do. Like, you don't really have to play around with it like too much unless you have some sort of more unusual data set. Okay, but Rachel's going to kind of go through those parameters when she talks about like the next round of search. All right, so here we are finally at the run task. We have databases up here, raw files on the left, and then all of the tasks. You can add as many as you want. They'll just run in order and pipe right into each other. Here we have just the one. And this is showing you where the data file is going to be. So I'm going to hit run, and you're going to sort of see real-time search. Ben, you see down here, this is the case where it's showing you there was conflict between the mouse database that we used to search and the contaminant database, and it's telling you sort of explicitly like which accession numbers are not being included as a consequence.
Okay, so you can, there's kind of different, schools of thoughts on like a vignette, right? But I mean, basically you're saying, with these files, you can pretty much go through, use defaults, and you're going to have results at the end. We're not going to be throwing errors. We're not going to be hitting these kind of more advanced problems.
That's right. And we provide, or we used to anyway, we used to provide the results of all of this. And so what you could do is you could pop open a couple of like Excel tabs and sort of look side by side, and you should get this same exact answer like every single time.
If you're running a like an Orbitrap, high resolution MS2 bottom up trypsin sample, you really don't need to mess with any settings at all. That's the defaults are sort of built for that sort of scenario.
And for, I imagine like someone who has no idea what we're saying, like what your words just said, like if you go and download data from Pride, those words should be there, right? Like it's a trypsin, it's going to say probably Orbitrap or high res, like that'll be in there somewhere. And yeah, so those settings are totally fine for MS. So a fragment ion high res type experiment.
Yeah, that's right. And we're going to talk about cases where you need to make those adjustments when Rachel gets around to, her section. So our search is totally done. You can double click on any one of these like files and it'll open that file directly, or you can hit the open button. And what open's going to do is just going to pop open the results folder. By default, you can see that it appeared up here in the same folder where the raw files were. So that's the default. Wherever you draw the raw files in, that's where the results are going to end up, okay? And it's named with the date and time, okay? And so you can search the same thing sort of like over and over and over again if you like, and it's not going to overwrite it. It's just going to like plop it down here as like a new time. In this folder, there's a couple things. I have a task settings folder. That's going to include like all of the settings that were used. So if you want to run the same search over again, or you wonder what the parameters were from your search, you can open this folder up and have a look at those files. Rob's going to talk about that kind of at the end. And then there's a search task folder. And in the search task folder, there's several files. We have the PSMs, the peptide spectrum matches. We have a peptides file, which contains the highest scoring example of every different PSM. There's an output. Some people like to run percolator on the PSMs after the analysis, and so we formatted a file so that you could read it directly. You wouldn't have to reformat our results if that's the kind of thing that you want to do. And also by default, quantification was turned on, okay? And so what we have here are two files for quant. One would be the peaks quantified, and one is the peptides quantified, okay? So peaks meaning like PSMs, and this would be like across files, and then peptides again was the example. And then there's one file that contains both the inferred proteins as well as the protein quant. Rachel's going to go through like all of the she's going to pop these files open and kind of like walk you through the steps. But I wanted to just show two additional files here. The first file is just it's a simple text file with the results. In this one raw file, the total search time was 1 minute and 6 seconds. There was 14,000 PSMs, 5,700 peptides, and 2,500 proteins. There's only the one file, so it tells you like how many spectra were analyzed and so forth. There was an XML for the contaminants database. There's some like listing of a few PTMs that we saw, but you'll see how that expands greatly when we do the PTM discovery in a minute. And that's basically it for like sort of a high level.
Hey, Michael,
yeah.
Hold on. So you had a raw file. Did we use the raw file here? So we've loaded a raw file, 25,000 MS2s, 1 minute. Like, what is your computer? Just give me like a perspective here. Like, do
Yeah,
you
let me
have
show
like,
you what the computer is here.
okay,
All right. So I'm
because
gonna
that seems really fast to me, and I know it is fast, right?
Yeah.
But like,
let me open up the pros file here. The pros file is made so you can kind of drag and drop it kind of into a paper. It has like just a condensed version of the parameters, like, you know, your sort of search parameters and stuff, but it plops in there your computer. And so this is a fast, powerful computer. It's got 28 threads and 128 gigabytes of RAM. On Rachel's like laptop, it's, you know, it's a good bit slower. Okay. You can sort of like run it in human time. You can certainly search files faster than you can take them, you know, but it's not like crazy fast, if that makes sense. It's like, you know, it's human fast. It's not, you know, our good friends at Michigan with MS Fragger, like we're, you know, they're sort of like at the next level with speed, you know, but this is perfectly reasonable sort of response time.
I just had to say that because, I mean, not that, like you said, I don't think it's unreasonable, but like that was really fast. I just had to ask, like, what are we doing here?
Search is also the fastest task. Calibration and Gptmd take a little bit more time.
Yeah.
Got it.
For my laptop, this would take about like 5 minutes.
That's still really fast.
So yeah.
Yeah, it's no slouch. I mean, let's just say that it's no slouch. But okay, cool. No, those files Rachel's going to talk about. Yeah, I'm excited about that.
I just want to, one last thing before I go is I want to show people the help over here on the left, because the way we feel about Metamorpheus is like, we're not going to be out there kind of like telling people like how great it is and, oh, you should switch and like use it. That's like not our deal. What our deal is like, is that for the people that are using it, we're going to like pour all our sort of time and effort into making sure that those people are successful. Okay. And so to that end, we try to make it like really easy for people to get in contact with us and let us know when things go wrong. Things do go wrong. Bugs happen, errors happen. You know, sometimes there's new features that people want. So first thing is that we have a wiki on the GitHub. That'll take you right to our GitHub page. There's a lot of information there that people can explore. And there's also a big glossary. So all the terminology that we use, like all of the different tabs that we have for like the data files, those are sort of all explained. There's a link here to the issues on GitHub. So I don't know if people don't know what issues are. Basically, it's a place where you can report on GitHub like that you're having trouble with some feature of the software. It's like not working for you. So you can open an issue on GitHub and say, you know, I tried to do a calibration and it didn't work for me. And we'll sort of see that right away. It sort of pops up in our notifications. There's also an email here, Metamorpheus support. People can email us directly and we'll get back pretty fast with that. And you can also find us on Twitter, and there's also some YouTube videos, and these will take you to links down there. So that's basically a variety of different ways that users can reach out to us and like get help in pretty close to real time if it's, you know, it's possible for us to sort of fix it.
And real fast, Rachel, before you start, just to so that, I mean, I found out about Metamorpheus. I mean, you know, all of us have kind of these closed bubbles, right? And, you know, yeah, it was, I think it was Brett was using it for his fall course. And his words were something like, it's silly how easy it is. The students love it. And I think I've now heard that from other people, right? You know, if you are trying to teach people that have never done any of this before, like what is it you're saying? Did you say like it doesn't require a computer science degree to use?
Even the PI can run it.
Yeah, even the PI can run it. And I think that's very underrated. So, and I think that's why this is really exciting for me, because, you know, I somehow got stuck in this old world because that's just how I came up. And this is great. So, okay, so Rachel.
So here, I'm not going to be doing a fresh install because that's already been done, but I have the same version of Metamorpheus open here. And I'm going to walk through a bit more of an intensive data analysis using the same vignette files that we saw before. Database, I'm just going to drag and drop in Uniprot, and this is an XML database. And so most people probably do grab their databases from Uniprot. Just go here. This is mouse. So
it's funny. I know most of the numbers. I do not know that number.
And we'll just look in the proteomes because it's a nice reference proteome. Here. And so when you're going to download your, database, normally you just click download and you can do FASTA, right? And that's what everybody's mostly used to, FASTA, either canonical and isoforms. But what we really like is the XML format, because this gives so much more information to Metamorpheus. More than we even need, because it has like all the publication references from each UniProt protein, but it has post-translational modifications annotated in it. So that's really a huge advantage in bottom-up analysis, but especially when we're doing work towards top-down proteomics. So we really like the XML database. Then you can either do reviewed or unreviewed, depending on how curated you want your sequences to be. So then you just download that by hitting go here, and that's the database that I'm using right now in its compressed format. So Metamorpheus can take compressed or uncompressed databases, so you don't have to wait as long for them to download. You can just, and you don't even have to unzip them once you have them there.
Hey, Rachel, can you crack open a FASTA and an XML and kind of show people like what each one looks like and why XMLs are cool?
And then, yeah, under format, yeah.
Okay. So here, XML gives lots of information. So it gives the accession, the full name, alternative names, genes, its entire taxonomic lineage, tons of references and all the authors on all of the references for like everything. So then we're just gonna like scroll for a while here because we don't really care about that right now. But what we do care about is modifications. So here, there's these features, modified residues. So it says, here's a phosphoserine at amino acid residue 10 in this sequence. So this is the information that we really like getting out of these XML databases, is all of these post-translational modifications that are there. It also provides information for splice variants as well.
And Metamorpheus for those modified residues automatically. You don't have to do anything extra. You can just drag it from XML.
And then here, so then for comparison, this is the same entry, FASTA. So bare bones, sequence, information about the accession, the gene, and its name. It's a much more pared down version, but there's no PTM information given in the FASTA format at all. So we're just gonna add in our Spectra, drag and drop. We've been doing drag and drop, but you can also just use add and it will open up a file explorer for you to select a file from as well. And then task window, all right? So our typical workflow will be a calibration task, PTM discovery task, and a search task. But as mentioned before, you can do a cross-linking search task if you're interested in using chemical cross-linkers. Or if you know you're having glycosylated proteins, you can use OPAIR for our O and N glyco searches here. So we'll start out with adding a calibration task. And so all of our parameters are collapsed right now. So if you're using, like we've mentioned before, just like a normal setup, you can not even worry about it and just click add calibration, but we'll kind of look at what some of these parameters are here. So in our calibration task, it starts with an initial search. So there's going to be lots of search parameters that we're doing here. It does a bare bone search to kind of figure out where we need to shift our peaks for calibration. And so it gives precursor mass tolerances, product mass tolerances, number of miscleavages you want to allow. You can choose what protease you want. So we have a list of like default proteases that you can select from. You can also add custom proteases to the protease file that's used by Metamorpheus. We have a relatively simple syntax to allow you to do that. So that way, if we don't already have that incorporated, it's not a big deal for you to add a protease to be able to search with. Providing minimum peptide length that you want to look for, number of modifications you'll allow per peptide, so on and so forth. We allow you to specify your dissociation type. So we're use CID, ECD, ETD, HCD, ETHCD, but essentially this all lets Metamorpheus know what fragment ions it should be looking for. And so let's leave that as HCD for now. And then max threads. So here, this automatically populates to 1 less than your computer has. So my laptop has 4 threads. So it's going to let me use 3, so I can still do things while it's searching. But for the purpose of when we actually run this, we're going to do some TV magic, and we'll shift back over to Dr. Shorey so that we have 27 threads running it instead of 3. Next, modifications. So here, this is where you can select any fixed or variable modifications that you want to be using. So carbamidomethylation, did you use iodoacetylene to alkylate your samples? Yes, make sure that's clicked here. Very rarely then are you going to want to select anything else in your fixed modifications. Variable, only one we have is oxidation of methionine, because we're going to do Gptmd, which allows us to identify post-translational modifications without a lot of the penalty of doing variable modification searching, of increasing the database and having higher false discovery rates with that.
Now, I know we're not going to go into that statement, but that's such a fun statement, right? Like, you're going to identify hydroxylated proline without having to kill your search space here. Yep. I know we're not going to go into that right now, but, or even
maybe there'll be a part 2 video and we can.
But I really, it's an understatement. You have a lot of understatements going on here, but that's it's a big one.
And then output options. So we write an indexed MZML file from our calibration task that will automatically be used for any analysis going forward. So we start with a raw file or an MZML file. It calibrates, creates a new version, and Metamorphis also automatically knows that that calibrated version is what it wants to use for Gptmd and subsequent search tasks. Add the calibration task. Next, PTM discovery. So here we do some, de-isotoping, peak trimming. This is just some general parameters for that. Again, typically you don't need to change any of these. Search parameters, same things that we observed in the calibration task. Gptmd again starts with just a search task. Same basic parameters. Same fixed and variable mods that we discussed before.
In the search parameters, those mass tolerances were automatically overwritten after calibration. So if you were to start with the Gptmd first and not do a calibration, it would use these values. But if you've run calibration, it's going to overwrite these automatically to whatever's appropriate for the file.
Yeah, we do file-specific parameters that are allowed to carry through, and I'll discuss that briefly in a moment. So Gptmd, so this is where you can select any modifications that you want to look for with our global post-translation modification discovery strategy. So here we have lists of common biological modifications that you might want to look for. So acetylations, methylations, some basic glycosylations, phosphorylations, etc. So we have this on by default. There's some less common modifications on some different residues. You can go through and select whatever you would want. There's not really much of a penalty for adding more mods, which is one of the great things about Gptmd. We have some metals that show up either biologically or can be artifacts of sample handling, and common artifacts as well that are typically due to sample prep, like water loss. We have those Gptmd mods, good to go, add task, and then search. All right, so this is like the final search task. So like I've mentioned before, calibration Gptmd internally runs some quick searches to allow us to calibrate and to do the post-translational modification discovery. But this is the search task that's actually going to be giving us our PSMs, peptide, go on to protein inference, all of those results here. So again, we have our basic search parameters. We also allow you to set the separation type that you're using. So you LC, or if you use capillary electrophoresis, you can do that here. And that's because we can calculate hydrophobicity or electrophoretic mobility. And this can be valuable in kind of like benchmarking your data and goes into the PEP calculation. And we have our modifications, fixed, variable. Then we allow you to say whether you want protein parsimony, protein inference to be applied or not. That's on as default. Our algorithm follows Occam's razor. We want to try to find the minimal set of protein or protein groups that can explain all of the peptides that we're observing. You can select for it to require at least 2 peptides to identify a protein or protein group. People have strong opinions on this either way. More evidence if you require 2 peptides, but now that mass spec is getting better, it's not necessarily wrong if there's just a single peptide. So use discretion. And then treat modified peptides as different peptides. So since we're using an XML database and we're doing Gptmd, we have this option of being able to distinguish proteins by whether or not they have a annotated modification there. So you can imagine you have a peptide sequence, and that sequence is shared between 2 proteins. Identification of that base sequence doesn't allow you to differentiate between protein A or protein B. But if in UniProt it says, well, this peptide sequence is phosphorylated, but only for protein B, and you observe this phosphorylated peptide, that can allow you to discern between those 2 proteins. And lower ambiguity is always, better, but you can decide whether you want these modified peptides to be treated as different when we're determining unique and shared for our protein inference results. That's off by default. And then we have quantification. Label-free quantification with Flash LFQ is on by default. You can switch to no quantification, or you can do some SILAC quantification. Match between runs, you can turn on normalized quantification results. Rob will discuss these more. And then we have a bunch of output options. So writing MZIDs, do you want your decoys written to your results files? Do you want your contaminants written to your results files? Do you want, if you're searching multiple spectra, to have a cumulative result as well as individual results for each file? Do you want these to be compressed to save space? And then finally, do you want to filter your results to a specific Q value? So this can be important if you're doing like a very large search with like 60 Spectra files. It's very easy then when you're opening the all PSMs file, that's going to be more than the 1 million lines allowed in Excel to open this file. So if you want it to be nice and Excel compatible and not have any issues and have to like filter using like WinGrip before you open the TSV, you can just set a results filter so that only the 1% PSMs are written out. And minimum score allowed. Advanced options, you really won't need to go into very often, if ever, but they do exist. File loading parameters, some additional search parameters, but a cool bit that I am going to discuss is some of this post-search analysis options that we have. You can construct mass difference histograms automatically, or you can output prune databases that you can use for future searching, which is something that we like to utilize. So here, you're going to create XML databases that are pruned to only contain entries that have proteins that are identified based on the spectra that you observe, and then only contain modifications that are observed or in the database based on the various settings that you do here. Now, it's important to note you shouldn't like search with files, create a database from that result, and then research those same files. That's like cheating. But what we've found can be really helpful is to do a multi-protease search, create an XML file that then we can use for a subsequent top-down analysis. So we're using bottom-up proteomic data to inform a database for a different analysis set. That's pretty fun. So we'll just add that search task. And then I mentioned multi-protease searching, and I'm just going to jump back here for a second. You can set file-specific settings. So you select a file or multiple files, and then you hit this set file-specific settings, and it says you can set this file is trypsin. And you can go to another file and say that file is from a collagenase digest or a lysine digest or a glucine digest. And so essentially you can take spectra from all different proteolytic digests and analyze them together inside the same GUI, because we know for each file how it should be analyzed with these file-specific settings. This format also is what enables us to be able to have file-specific tolerances that are carried through based on our calibration results. You could also do the same thing for separation type. So there's a variety of different settings that you can have be specific for individual files, which can be very beneficial. Those are tasks that we're going to be doing here. And again, summary run window, we'll have each of these displayed in the order that they're going to be run. And at this time, we're going to kick it on over to Dr. Shortreed, who is going to start the actual analysis.
Is this the magic of TV?
Yeah.
So you see here, I have exactly the same 3 tasks set up that Rachel just set up. And the reason that we're gonna like hit run here and not just like bop to the results is so you can kind of like, so this is gonna give us like about 5 minutes to kind of talk and chat between ourselves. But occasionally we're gonna bop back here so that you can sort of see what's going on sort of in real time, if that makes sense. So I still wanted to show you guys how the search is working. You know, in some compressed time, but it's gonna, like I said, we're gonna have about 5 minutes to talk here. I'm gonna queue this up, and you're gonna see like it's gonna go through each of these tasks. And some of the things we're like watching for is some changes over here. So we should see a new file show up here with the tolerances, and we should see some new databases show up here that have like PTMs in them.
And then just to kind of, so Dr. Shorey said this is going to take about 5 minutes on his. If we were to run it all the way through on mine, it's going to be about like 40 minutes on that. So again, not unreasonable, but just for the sake of trying to keep this video concise and get as much information out there as possible.
Yeah, no, so now if you had just done just the search only, so no calibration, no Gpmd, it would still be like how much quicker would that be than like on your laptop? Not
so it will be about like
speeds.
5 minutes on my laptop for just the search task. Calibration task takes 12 minutes and Gptmd took 24.
Man, you're on it.
Yeah.
All right. Yeah. I mean, one technical question and then one bigger question. So is it every time you run the calibration task, you're going to get file specific parameters? I mean, let's just hypothetically say I had 35 different instruments running a sample, all raw files. I could throw those in here. I mean, I would have to still fill things out, but if I had the calibration, each file would then, you know, let's say this person's running OTIT, or this person's running a TIMS, well, whatever. It would go through and give me different settings.
Yep.
Okay, that is crazy. I mean, so not hypothetically, you know, because we do these multi-lab studies, right now we just collected all this data, and I do have 35 instruments. And for us, we, you know, do you take what a user reports? Is there, you know, what they think is good? Another valuable thing here is even, you know, let's say you're 2 weeks out from your calibration for whatever reason, and your thresholds are getting a little wonky. This would kind of adjust for that, right?
That's right. Usually we use it like with a whole group of files from the same machine. We're not like in our lab, we're doing like, you know, like experiments, and all the data is run on the same machine. And so every file is going to get its own tolerance, and they're usually going to be quite close. But you're right, Ben, if the sun comes out and the light shines on the mass spec, you know, like the calibration can change a little bit, or, you know, after a couple weeks, there's some drift.
So the way we estimate that is the calibration task performs a search, and then there's some actual calibration in there where it tries to find a spline for ppm errors and stuff across retention time. But in terms of like calculating like the recommended ppm tolerances, there's a search after the actual calibration part, like a post-calibration search. And then so you get a distribution of ppm errors that's kind of like normal-ish. It's a little bit sharper than normal usually, but normal-ish. And so we calculate the interquartile range of that, which is like the middle 50%, and then multiply that by 4. So it is a statistical measurement of how broad that distribution of ppm errors is. Mm-hmm. If you actually look at the calibration task output, so the output is MZMLs, but there are also some additional text files that tell you sort of the pre-calibration, median and interquartile range of that ppm error distribution, and then post-calibration. So if you're comparing instruments across labs, doing an inter-lab study, that'll help you kind of figure out, is someone just like systematically off, but like they have a tight range, or someone's like errors like super broad or whatever.
Another nice thing is that like once you calibrate a file, then you don't so if you're going to repeat the search because you want to try a different Gptmd setting or something different with the search, you don't have to recalibrate that file. You can then just go straight to the calibrated file that we output and start from there. So like your first search might take longer because you're doing calibration and Gptmd and search, but then you have your calibration and then you just need to go Gptmd and search. Or if you're happy with your Gptmd, then you just need to grab that database from that output, your MCML from our calibrated search, and then just redo the search analysis. So it's designed to be as easy as possible to make repeat analysis fast and efficient.
Not only is like the file like calibrated, but the calibration, the file specific parameters are stored right next to it. So as soon as you drag that MZML file into MetaMorpheus, it's going to look and it's going to say, is there a calibration settings here? And it's going to grab that automatically. So you don't have to like remember where it was or what they were. They're just going to go right back. And the same is true for proteases. So if you combine a trypsin run with an aspen run, it's going to remember that those files were trypsin and these other ones were aspen, and it's going to just sort of drag that in for you. And here you can see sort of already there's a couple things that have showed up, okay? Down here in the bottom, you can see we have a brand new entry, and it shows you what the precursor and final product mass tolerances arrived at. And now this box is checked and the first one's unchecked, okay? And so it used that subsequently. And up here, you can see there's two new databases. So what Gptmd does is it doesn't do the search. It creates a database of putative PTMs based on mass differences between experimental precursor and theoretical precursor. And we don't really have to get into too many details, but we started with an XML with some annotated PTMs from UniProt. And then Gptmd added a bunch more PTMs on specific peptides where it thinks that there's reasonable evidence for that. So now these are checked, and the same is true for contaminants. And so the new file and the new databases were used in the final search task. So everything's done now. And I'm going to switch back over to Rachel in like 1 second, but I only wanted to open up like the basic results file so that people can sort of see like a more complicated results. So again, this was like 6 minutes on my microwave oven. And so, there's some statistical parameters down here that have to do with like a little machine learning thing that I tacked in here. But I want to scroll down here. These are the modifications that Gptmd added. That's just kind of a laundry list. You can see kind of really common ones like deamidations, quite prevalent. There was some sodium adducts in this sample, so it like automatically kind of looks for those. And I think that's kind of key, if you're trying to really keep track of all your PSMs and so forth. But you can see some phosphorylation showed up in this like single file, some acetylations and so forth. And then if you scroll down here, it'll actually tell you specific like things that were actually found. So those were mods added. And then here are PSM counts for the various modifications. Okay.
And can you, so when you say like they were added, like above, I mean, those are, again, you're not talking, we added these to my search space, like the whole search space. You're just like, look, this right here at this protein, I think there could be a sodium adduct. Yep. Check it out.
That's right. So in the whole database, we added 347 deamidations on asparagine only. Like, so it's a really tiny number. If you had like counted all the ends in the entire proteome of mouse, there would be like thousands. And Metamorpheus found 347 possible spots where a deamidation could have occurred. Okay, and so that's why there's not a database explosion. It's like super limited. Okay, and then the search is really what refines whether we think that there's really a PTM there or not. Just because we added the possibility, like it still has to kind of kind of wash out at the search level. Show you those sort of same results, but she's going to go through them in a lot nicer detail and also show some great visualization tools that we have.
Going here, I did the exact analysis just before we got on here. So we have our results files. So calibration task. Here is the calibrated file that you can use for subsequent searching. This .toml file is all of those file specific settings. It's calibration parameters, and if you created a file specific protease. Pros and results just give some additional information on those files here. Gptmd, it has our new database that we would use for our subsequent search, and then also gives you a TSV file with all of the various PTM candidates, kind of what was summarized in that all results file that Dr. Shorey shared. And the rubber really hits the road is here in our search tasks again, where we have our all peptides, PSMs, peaks, and protein groups. So I'll just start with our all PSMs file, and then it's going to open an Excel for me because during the installer portion, I click the little checkbox that says make Excel the default opener for these file path types. If you don't do that, then you can just drag and drop that file into an open Excel document and it will bring it up. You can also open it in like Notepad, but it's much prettier to have it put in all columns, and then you get all the filtering capabilities of Excel. So here we have the file name, we have the scan number for each PSM, its retention time, number of experimental peaks observed in that spectra, the ion current for that specific spectra, precursor scan number, charge, MZ, the mass, and then we get into score. So the Metamorphis score is like two parts. There's the integer part and then the decimal part. The integer part is the number of matched fragment ions between the theoretical database and the experimental spectra. So for this first one, 28 fragment ions were matched. The decimal portion is the percent of the total ion current that is occupied by the peaks that we've identified. So 31.6% of the total ion current for this spectra is accounted for by these 28 matched peaks. Delta score is the difference between, for each PSM, the top scoring match and its next best option. So high delta score is good, meaning that there's high confidence in what we have. So the next best score would have had like 5 matched ions, where this has 20. Notch, which is for if this was going to be like modified. We have our base sequence, which is just the amino acid sequence itself. Full sequence will contain any post-translational modifications. So here, the next one down has an artifact that we discovered with Gptmd. And so that's annotated in the full sequence. The essential sequence is a like pared down version of the full sequence that's more curated. PSM count here is kind of like, gives you the spectral count for that specific PSM. So how many other same base sequences, unambiguous identifications below 1% FDR are there for this. So for this PSM, this base sequence, we have 6 other PSMs that support that there. Then we have some columns to show modification. It's chemical formula for that mod. If there's more than one modification on the peptide, what's the overall chemical formula combined for those? Then we keep a total of the number of variable modifications for that peptide, the number of miscleavages for that peptide. We give you the monoisotopic mass, its mass difference between theoretical and experimental in Daltons and ppm, protein accession, name, gene name, organism. If you use a Spritz database, which is another tool that came out of our group, which creates sample-specific databases using RNA sequencing information. This will let you know if the PSM or peptide that you identified has a identified sequence variation within it, or a splice site. Tells you if it's a contaminant, if it's a decoy, if it is like a full, so it's like if you allow, like if you do like a semi-triptych search, it will tell you if it's like a full triptych or a semi-triptych peptide. Start and end residues in the protein, the previous amino acid, the next amino acid. Then this is another way to filter target decoys, contaminants, all of the matched ions that we have. So say like, oh, look, we found the B1 ion in charge 1 state, the B2, B4, so on and so forth. Gives you the mass for those. Also will let you know if there's any like neutral losses, the match for the mass difference for each of the mass ions, in ppm and Dalton, the intensities for each of the matched ions, and then the count. So here, right, like I told you, there was 28 because the integer of the Metamorphis score. Well, here's another way. It's 28 just listed out here. Normalized spectral angle, locus scores. Okay. Here is where we get into calculating target decoy for FDR. So we have our cumulative target, cumulative decoy, and then Q value for each PSM. So you can do your global cutoff. And then there's cumulative target notch, which allows you to have the FDR specific to the modifications that are discovered. Cumulative decoy notch, Q value notch, and then the PEP value that we can calculate. So all these column headers are the same between the PSM and the peptides file. The difference is going to be in the PSM's file, it's all the spectra matched. In the peptides file, it's only the best PSM for each spectrum. I'm going to jump to the protein group file. So this is only made if you do protein inference. And so post-protein inference, we create proteins and protein group identifications, and those are also then mapped back to the PSM and peptides file to make sure that everything like corresponds. So we have the protein accession. So here, we are able to determine that this single protein is in our sample, or this is how we display protein groups. So these 3 protein accessions, we can't distinguish based on the peptide evidence that we have, if one is in the sample, if it's a subset, or all of them. So we display all equally here. And this is the shared peptide support for that. So here we give our protein accession or the protein group accessions, gene, organism, names, the mass of the unmodified protein, the number of proteins in each group. So this is a nice way to kind of filter there. And then we tell you which unique peptides we've identified for that protein and which shared peptides we've identified. Total number of peptides, number of unique peptides, and then sequence coverage fraction. So we say, based on the peptides that we've identified for this protein, what percentage of all amino acids do these cover? So that here, and then here's some like kind of way to visualize that. So we have sequence coverage here. Lowercase are that are not covered. Uppercase are ones where we have peptide support for that. It gives it as a base sequence. Also, if there's modifications, it includes that here. Gives the intensity for that protein, the number of PSMs for that specific protein, whether it's a target or decoy, the best peptide score for that protein. And then we do a protein level FDR that is kind of based off of the top picked approach. So, which essentially means that if you have a target protein group and a decoy protein group, and the target outscores the decoy protein group, you keep the target protein group, which helps not be overly conservative in the estimation of protein level FDRs. And then, as mentioned, I am going to show you some nifty visualization that you can do in Metamorpheus /MetaDraw. I just opened up the desktop icon for Metamorpheus, and I'm going to click the Visualize tab here, and it opens up the MetaDraw window. So I'm going to select the Spectra file. And so since I did calibration, I need to select the calibrated Spectra file for this. The PSMs. And then load. So this is going to give you spectral annotation. So here you can just click and you will get an annotated spectra for all of the different PSMs that you identify with some nice information like what was the precursor charge, the precursor mass, its score, so on and so forth. So blue are our B ions, purple Y ions. So you can see annotated spectra here, and then you also can see the nice little stepwise graphic on the sequence as well. You can also do search here, which is really nice. So if I really want to look and investigate, like what did the spectra look like for phosphorylated peptides? So I can just type in phospho. So here, click on a phosphorylated peptide sequence. And so there's this yellow dot that covers our modified residue. So then you can easily observe how many fragment ions do I have that would cross this phosphorylation site. And then again, you get to see the annotated spectra with the neutral loss annotated as well, which is great. So this is a nice, easy way to do your spectral annotations. And then data visualization allows you to do some nifty plots. So like histogram of precursor ppm errors, histogram of precursor charges. You can look at the spectral counts for the various post-translational modifications identified, or look at predicted versus observed retention time. This one takes a little bit longer to load, but then it's better with you if you use multiple like files, but you can see like there's pretty good, you know, trend here of these are likely our nice high quality identifications. They trend well between predicted hydrophobicity and what we observed in retention time. So all of this is right at your fingertips inside of MetaMorpheus. And then here, I just want to note, so this is like non-variant PSMs. If you use that Spritz database that I mentioned and you load in and you do MetaDraw, there's will be like purple dots that are for the variant containing PSMs. So then you can see, do they behave the same as the normal PSMs that you're observing? But just want to specify that because it's kind of weird to say non-variant if you don't know that there could be variant ones there. So you can export all these to PDF as well. So you can export any of these graphs to PDF and any of the spectral annotations to PDF. I'm done and I'm going to hand it off to Rob so he can show you the wonderful world of quantification.
Oh, yeah, Rachel, that was great. I mean, I feel really bad because every time you show me something, I'm kind of like, what? Like, I mean, I feel bad that I haven't been using this, but even like the visualization, right? I mean, that's I mean, is that something that's just always been around, or is it like a newish feature? I mean, I know some of these things are newer than others.
I have like lost track of all sense of time, like since the pandemic started. So like my initial gut instinct was like, it's been around a year, but then that's totally not true. So I think it's like 2.5 years that we started like incorporating MetaDraw into our analysis. Yeah.
It's still really cool. No, that's, I mean, it's, yeah, I mean, I won't go into it. It's very mature, right? I mean, all these things that you're kind of building. So
yeah, we want it to be as user-friendly as possible and kind of like one-stop shopping if possible too, because it can be difficult and annoying to pipe results through multiple different tools to try to get an end result. And so
I don't know what you're talking about.
Right? So a lot of this is just like came born out of like frustration of stuff from our group, like wanting certain results. And then as our group became empowered to be able to create software tools, we have an idea, let's incorporate this into MetaMorpheus or into our suite of tools so that like if we want it, sure other people are interested in this too. And how can we make it as easy as possible for everyone to access and achieve that?
Some of it too is kind of hidden. We recently redid the GUI to make some of this stuff more obvious and to make a more kind of linear workflow also. So yeah, a lot of the visualization stuff used to be kind of hidden in menus, and even now the current MetaDraw is sort of in the old style, and I think the data visualization stuff is kind of hidden relative to the kind of the spectrum annotation. So I kind of want to make that a little bit more prominent. But
well, now that we've made a video, you can't change it. So
yeah,
sorry.
One other point, Ben, that I wanted to kind of bring up, like, you know, I mean, one reason for sure is that it's like nice to have all of this like sort of stuff handy, but like I'm like by nature kind of skeptical of like randomly assigned like PTMs or like amino acid variants. I think sort of as a community, like we have to be like, you know, like it's fine to run a regular proteomic search and say you saw this or that thing. But if you're going to say like I saw phospho here or a acetyl there, or there's an amino acid substitution, I think the bar really is a little bit higher for that. And we have to kind of do the work to say, that we stand by this assignment. And so having a tool that can annotate the spectra is kind of step one in the sort of manual validation landscape, when you're talking about like kind of going to the next level with that, especially like with Gptmd. You saw the different variety of mods there. Like if you're not skeptical, you should be. And so like, you should be able to have at your fingertips a way to kind of manually assess those things. And so, we try to build that accessibility in so that people can do the right kind of scholarship in order to report these things.
Right. Yeah, no, and the whole open searching has been around even, back in the day with Mascot error tolerant, right? Like, but yeah, there's a grain of salt required. And I think, yeah, the approach and the validation and being able to like look at it is key. So that's really cool.
Michael, do you want to just talk about the data set we're going to analyze?
Yeah, absolutely. Let me bring it up here quickly. So what Rob's going to show you isn't like a sort of a complicated like biological experiment. What we're going to show is a spiked in yeast in a human background. Okay, this is a publicly available data set. This was produced by the lab of Steve Giggy at Harvard in a paper where he did a comparison between label-free quant and TMT quantification. Okay, but it's just a really nicely made data set on high-res tandem aspect with trypsin, and so we downloaded that. And so it's just a chance for us to show you how to set up a quantitative experiment and then how to like visualize the result. Do you want me to say anything more about that, or is that kind of cover it? And again, I think what we'll do is like after the video, when we post this, is we're going to post some like links and discussion below so that people can get access to this paper and this data. Like I said, the data is publicly available.
Yeah, I think that covers it. Like Michael said, we're going to analyze. I've already actually analyzed this data because I'm also on a laptop. This would take a few minutes to do a search and quant, but I'll show you how to set it up at least. So I'll start Metamorpheus. I deliberately installed an old version. And the reason for that is I want to show you how our auto update works. So if you run an old version of Metamorpheus while you're connected to the web and you have auto update enabled, then Metamorpheus will try to find or try to detect if there's a newer version of Metamorpheus that you can update to. So when you run it, it'll check, and then it does actually have a new version 0.0.315 technically. I'm going to say not interested just for right now, and then also show you how to update via settings or via help, sorry. So you can also manually, if you have auto update disabled or whatever, or if you just want to check for updates, you can check for Metamorpheus updates there, and you can download and run the installer. So it'll download in the background, download it pretty fast, and then sometimes Windows kind of freaks out for a second, but it looks like it is okay. All right, this is where it tends to sort of hang for a second because Windows is freaking out about security stuff, but okay, we're all good. So that's auto update. So now I'm on 315. I'll just drag and drop these raw files, and you can drag and drop from any screen. So I'm not even on the Spectra screen right now, and I was able to drag and drop all of those. And then I actually don't have a database on this laptop, but that's fine. I don't actually need it to set this up. So these raw files are a little bit confusingly named. They're not really indicative of what is in the sample. But the way this is set up, this data set is set up, is you have a set of files. They each contain human and yeast lysate mixed together. Each sample has human and yeast mixed together. But the yeast amount is in different like amounts depending on the file. So there's three sort of quote unquote conditions. There's the 1x, which is sort of like the baseline, this like amount of yeast. Then there's another set of files where it's double the amount of yeast lysate, and another set of files where it's triple the amount of yeast lysate. So you should see the amount of yeast intensities going up, but the human should stay constant if quantification is doing its job properly. And so the way you actually don't have to set up what this experimental design to do a quantitative analysis, but it's better if you do, because it lets you do normalization, and I'll explain what that is in a second. But you can I just memorized what these files are, but this is part of the 3x condition. So it's 3 files in the 3x. There's 4 files in the 2x, and then 4 in the 1x. And then what you can also do is if you already have this kind of set up, you could copy and paste from Excel. So I'll do that. So I'll just copy paste it into there, and that takes in comma delimited or tab delimited cells. So the way this is set up is for each condition, you have to have sequential, basically sample numbers, biological replicate numbers, starting with 1, and then they have to be sequential. And then, let's say you have fractionated data, right? If you have 1 biological rep that's fractionated, you would, let's just say these are fractions of this sample. You'd have fraction 1, 2, 3, 4, and you could have technical replicates too. So it's sort of almost a tree structure, if that makes sense. But this is not fractionated, so it's not as complicated. I'll copy and paste mine in. And if you do mess something up, let's say you accidentally put in like 4 or something, and you try to save it, it'll tell you biological rep 2 is missing in condition 3. So it was expecting a 2 here. The condition can be named anything. It's just a string. There's no format needed for that. It's whatever you want to call it. But these all need integer numbers. So save, it'll check obviously to see if everything's valid, and then there was no error message. So at least unless we mislabeled things in a acceptable order, then it's okay. So so I've got my experimental design set up. I'm also going to create my search task, which includes quantification. So match between runs will try to find peptides that weren't fragmented and identified in other samples. So what that means is, like, if you have one run where a peptide was fragmented and ID'd, and let's say in another sample it wasn't, the peptide is in the MS1 space as a chromatographic peak, but it wasn't fragmented. Flash LFQ will try to map this peak onto that peak and say this is the same peptide. That's what match between runs is. And that uses retention time alignment between samples. And then normalization is an intensity correction, basically, where there's typically some drift in measurements between samples. So like if you run 3 samples in a row, even the same sample, even if you're in the same sample 3 times in a row, the instrument can drift a little bit, and the intensities can decrease over time. And the other thing is that if you're running even seemingly identical samples, but they're different samples, there can be sometimes very slight systematic differences in how much like total lysate you inject or whatever. So normalization basically just finds if you're looking at 2 files that are in the same condition, or 2 biological replicates that are in the same condition anyway, it'll subtract peptide intensities between those 2 files, find the median difference, and set that to be 0. So the assumption there is that even between conditions, the assumption is that the median peptide is not changing in abundance between those samples. And that's a pretty common assumption when you're talking about normalization methods, but we use median centering. So I'm not going to run it because I forgot to download my databases, but I also have it already done. So I'm going to close this. Do you have any questions about setting that up?
No, I mean, this is, you're explaining it well. You're hitting things that I wouldn't have even asked, like whether or not it was median or mean centered. So yeah. I mean, I appreciate that.
Yeah, I tend to use median for a lot of things because it's just much more robust to weird tails and stuff. But I guess I should also say it when you save your experimental design, it will save it as a TSV file. I've already got it open. So you can look at the various files and then the condition and biorep, et cetera, et cetera. When you do calibration, it automatically updates those names with the calibrated MZML name for you. Okay, so I'll show you the quant output. So let's look at peptides first. That's pretty simple. Okay. So here we've got the peptide sequence that was quantified, and that tells you also what the base sequence was if this was modified. So for example, here's a carbametamethyl C modified peptide, but here just displays the base sequence along with this protein and gene name. And then you can see the intensities of the peptide across files. And you can see so this one's probably a little messed up. You can see this is supposed to be a yeast peptide, so maybe this one's pretty noisy, but there should be a sort of a systematic change there, but the proteins are a little bit more obvious because there's more data per protein than there is per peptide. And this tells you if the peptide was quantified with MSMS, meaning it was fragmented in that file and the peak was found, or if it was quantified with match between runs, or if it just wasn't detected at all. There are also some errors where if multiple peptides find the same chromatographic peak, Flash LFQ will just cancel out that peak and says don't use this for quantification. Something's weird with that peak, and that happens a lot with phosphorylated peptides. Is if you have a peptide that's phosphorylated here versus here, they'll kind of collude. It's hard to distinguish those. If you want to look at the peaks, this might take a little while to open. It's kind of large. So this tells you the actual chromatographic peak information. Usually this isn't really necessary to look at. It's just kind of interesting if you're looking at, if you're interested in the chromatography. It'll tell you where the peak starts. The apex where the peak kind of crests, and then where the peak ends in the MS1 space. And then some other stuff like how that peak was detected. Also, I should say how Flash LFQ kind of works is, so Metamorpheus will feed in the PSMs that were reliably identified into Flash LFQ. And then Flash LFQ will start at that MS2 fragmentation event and then look around that place for the MS1 peak. So this PSM's mapped column, what that means is this peptide was fragmented, for example, twice, and both those PSMs found the same chromatographic peak. Whereas this one, let's say, was fragmented 11 times and one peak was found where all 11 of those mapped to the same peak. So there's no double counting or whatever going on. There's some ppm error and other interesting measurements there. And there's also some ambiguity when, some PSMs find the same peak. So here this peak was ambiguous between this peptide and this peptide. And then probably what people usually care about is the protein groups file. So here, this is a quantitative experiment, obviously. So we've got multiple intensities. And so these are all human at the top here, but if you scroll down a little bit, you'll find a yeast file or yeast protein. And if you plot the intensities across the files, you'll see that there's three files that are sort of high, 4 that are sort of moderate, and 4 that are low, which matches what we'd expect from our experimental design. So the quantification, at least for this one protein, looks pretty good. And there's obviously some noise in between. So typically what you are looking for in a quantitative kind of LFQ context is what's called a fold change. So how much a protein changes between samples. That's because this is a relative quantification. It's really not an absolute quantification. So I won't really go into that much, but so if you, for example, take an intensity in the 3x condition and divide that by an intensity in the 1x condition. So this is human. This is a human protein. That's why it's not. That's why it's about 1. So it actually should be about that. You find a yeast protein. Just highlight this. And then like this, for example, divided by that, it's around 4. So again, around 3 block with some noise. So that can result in some pretty large numbers or very small numbers. So typically these are log transformed just to make them a little bit more easy to plot, really, is what it usually is. Log transform that, log transform that. This is log base 10. I usually use log base 2, but it's a little bit more intuitive. So this minus this will give you a log fold change. In this case, it's log base 10. And because Excel can sometimes can mess up a little bit, you can give these error messages if there's like no measurements, because sometimes there are missing measurements, so missing values. I'm going to change this a little bit and say if there's an error. Input A. And that'll let us plot it in Excel properly. So we can make a little histogram. And you can see like there's a peak around 0. Those are the human proteins. And there's a peak around log base 10 of maybe 0.4 or so. And that's the yeast proteins that are changing. So another cool way to look at it is you can plot it. I've already pre-made some graphs. Here we go. So there we go. So the x-axis is the intensity of the protein, and the y-axis is the difference in intensities between the two conditions for various proteins. And so the black dots are human proteins, and the blue dots are yeast proteins. And it's around, this is log base 2 of like 1.58 or something like that. That's around that's a 3-fold change. And you can see at lower intensities, there's more noise. That's just a signal to noise thing. At higher signal, there's less relative noise, and at lower signal, there's more relative noise. So I think it's kind of a nice way to visualize this. Of course, in a real sample, like if you're analyzing cancer, for example, if you're doing like tumor versus no tumor, it'll look more like the black, and then there'll be a couple dots out here that might be differentially expressed proteins. You won't get a nice, separate distribution in real life when you're analyzing these real samples. But this is just kind of showing that the quant does actually work. That's why the spike and stuff is nice. You can kind of tell which proteins are incorrectly quantified and which ones are correctly quantified. And so we use Perseus a lot to do like t-tests and stuff. So you can actually import flash LFQ results directly into Perseus, but you can do a generic matrix upload from our TSV, our protein groups TSV. I won't go through all the t-testing and stuff, but so this isn't really a Perseus tutorial, but you can add the intensities to the main thing. I'm gonna get rid of this. You can add the organism to the categorical, and then the protein and gene to text. And the reason why I made organism and categorical in this case is because there's 2 categories here. One's yeast, one's human. Yeast changing, humans not. So if you want to label stuff like I did in the previous chart, it's a little bit easier to make that a categorical thing. So that's it. That's all you have to do to upload data into Perseus, and you can log transform and filter by valid values, because there are some down here that have quite a few missing values, and then you can impute and then do t-tests and stuff. And I can go through that if you want. It takes a couple minutes, but it's not too bad. And then with this data set, I think we at an FDR of like 5%, I think there's actually like maybe on 8% or so that are significantly changing, that are human, so they're incorrectly changing. But the FDR control is pretty good for like t-tests and stuff. It's not amazing, but it's not bad. So that's kind of like the basics of quant, and like some cool charts you can make. I'm gonna move on to the next thing if that, you have any questions about that?
No, I mean, as a person who does a lot of label free, just seeing that it spits out that Excel, right? Or that, that matrix. I mean, I can do whatever,
Yeah.
but I think to people that haven't been around very long, you think that it should just make that. But if you just go back 10 years, even, PD wasn't even around 10 years, it wasn't making those graphs or that table. So, I mean, that alone is just good to know that that's an output.
Yeah. Okay. I guess I should note that older versions of Metamorpheus, we used the top 3 protein which wasn't that great. We've since switched to median polish, which is, I think, pretty good. I think it's about as good as max LFQ, but it can handle more data. It can compute stuff faster when there's a lot of data files. So I'll talk for a second about TOMLs. So this is a TOML for a task that I ran last night, as you can tell by the date. So the TOML file isn't really like especially it's not like some esoteric mysterious file format. It's actually just a text file. So you can open it in Notepad and then read like what these different settings are that we use. There's a lot of them, but like for example, if you wanted to change the ppm tolerance for quantification, you can make that 10 or whatever. So what's really, I think, really useful about this is you can pass like how you ran a task to someone else, which is really good for reproducibility. Like if you publish something, it's really important that they be able to recreate your experiments. So you can just pass, these small TOMLs and then obviously the raw data and databases and things, and you can recreate or run a Metamorpheus pretty easily. You can just via drag and drop that creates a task. So
yeah, the TOML files are for each search task are created like once you say like add task and like you start the run, then it saves those. So all the parameters that you set in the GUI are then saved to that TOML file.
Yeah, so there's no tasks. You can drag and drop search task created, and it's the same settings. So yeah, like Rachel said, when you click run, it'll create this folder task settings. Another option, if you don't want to run it, you can right click and click save as TOML, and it'll give you a path. You can save it wherever you want. So typically, there's also a command line version of Metamorpheus that I'll show you in a second. You can create TOMLs with the command line, but personally, I would recommend if you have a Windows machine, I would recommend that you create the TOML via GUI and then save as, and then use that TOML in the command line, because editing these TOMLs, you can break stuff. Like if I made this decoy type to be like something else, like Metamorpheus would not run properly because that's not a valid like decoy name. So just to make sure you're doing stuff right, I think it's usually better to make stuff in the GUI and then transport that to the command line world.
Here, I'm going to ask a question while we're doing that, because I didn't ask anything about you guys. So as you're preparing your command line, your CLI fun, how many people are in your group?
15.
Michael, you or whoever, yeah. Anyone, 15?
Fish, 15, 12.
And is everyone in the Metamorpheus universe, working on all these different, okay.
We have like, I don't know, there's like people who like informatics, there's people who are only in the lab, and then there's people who like live in both worlds, which I am one of those people. But yeah, it's great because we like automatically have like users to test that like don't program, and people who don't program can come to us with ideas and suggestions, and we can like try to make it a reality.
So, okay, so like 15 people. And so like, let's say all of you are at ASMS and you show up and like, there's these other people from Michigan. Do you guys have like a Rumble? I mean, I know that you don't, but I wish that you did, kind of like a West Side Story. But no, seriously, I mean, that rivalry, it's not a rivalry, but I feel like
it's a friendly rivalry.
A lot.
Yeah. It's,
yeah.
I think it's really cool.
Twitter, you'll see things like if Michigan beats like Wisconsin in football, it's possible that the Michigan group will mention that to us, or whatever. But the truth is like sort of competing with those guys and other people in this field. Like honestly, I don't know if there's like something special in the water with the proteomics community, but everyone's super cool. Like, and people will share how they do things with each other, right? It's like I mean, we want to compete in the sense that we want to drive the field forward. And of course, kind of like our pride says, like, you know, we want the best at like whatever. But at the end of the day, we're just trying to do like really good proteomics. And so I think we're all quite close. Like, honestly, we'll go to each other's posters and we'll share like everything we like know and learn. And it was funny, like when we started making Metamorpheus, like my boss said, like, why do you want to make a search engine? Like, can't people do that already? Yes and no, right? Searches could be done with FASTA files at that time. You could identify proteins, 10 years ago, but there's so much more to be learned. And I really sort of feel like with proteomics, the same thing is still true. Like proteomics as a field has evolved a lot in the last decade. There's like lots of cool stuff you can do that you couldn't do before. But I feel like there's just as much fun stuff to do now. And those are the things we kind of like continue to discuss and argue about. And like, we're not sure we have the right answer. Top-down's a perfect example. Like our ability to do comprehensive top-down proteomics experiments is just not good enough, as a field. Like we're just kind of skimming the very top, most abundant proteins, very low molecular weight. We are not doing comprehensive top-down proteomics. There's a whole bunch of work to do. And so, we can go to meetings and we can haggle and argue about how to like attack that problem. And there's good competition yet to be had.
No, that's one of my favorite things. I mean, we're not getting into all the other things that Metamorpheus does, but I know that you guys have your eyes set on these other targets way beyond the things we're talking about. I mean, that was the point here, is to start very simple. But yeah, it seems like your group, I mean, it's as a developer team, or whatever you want to call yourself. I mean, how do you have this lasting power? Because I know lots of people will come up with an algorithm that is supported for 2 years, 4 years. Right.
So one thing that we did, which is different from a lot of groups, is we try to keep the number of programs like really small and add functionality. Okay. So we're doing cool things like Flash LFQ, and Flash LFQ runs by itself, but it's actually like also in Metamorpheus. Okay. Like the glyco search is it's in Metamorpheus. And so what that means is that we're all working together continuously over time, and the tool's evolving. And it's not just a one-off thing that kind of gets lost when the grad student graduates. Does that make sense?
Yeah.
So, grad students kind of want their own project. They want to publish under their own like software tool name and stuff like that. But what happens is, as you know, you kind of have to recreate the wheel every time, right? So if you're making a thing to do a cross-link search and it's brand new, you got to learn how to read a raw file again. You got to be able to read a database again. You got to be able to do all the error correction, okay? So we worked hard at the start, making all that stuff work in Metamorpheus. So now when you want to add crosslink, it already can read a file. It already can read a database. It already prints out the results. It does all those things already. And then you can focus on the smaller task of adding the functionality, okay? And it's got to work in the whole. It can't crash everything. It's got to be like bug free. And in this way, so as long as we continue to add functionality to Metamorpheus, it will live, okay? When we stop adding functionality and start spinning off in like different things, that's when things kind of go crazy a little bit, I think.
Right.
Another part that's helps with that is the fact that we all review each other's code before anything goes in. So if someone's working in an area that you're not familiar with, you're reviewing that code. And then in the effort to make sure that nothing breaks Metamorpheus, you're learning what that does, and you're learning that area, the glyco search that you didn't develop, but you're helping review that code. So now you're understanding what's going on there. So then when people graduate and move on to different projects, there's at least some level of understanding that stays within the group to help continue things moving forward.
Yeah, that's really cool. Really, really is. All right, CLI.
Yeah, let's do it. All right, so I have a command. So this is a Windows command line, but you can also run it in Windows and Linux and Mac. Kind of explain that in a second. But so I've just navigated to the. Well, actually, I'll show you. I downloaded the Metamorphis command line.zip. I extracted that to this folder. I've navigated to the folder in the command line interface. All you have to do is just run the cmd.exe. It'll spit out that you didn't use any flags. You use the help flag to show what all the other flags are. So the most important one, or three most important ones, the TOML flag, the database flag, and the Spectra file flag. You can also optionally specify an output folder, and then you can also generate the default TOMLs if you want, so you can edit those, generally speaking. But yeah, I already talked about, you should probably use the GUI to generate those, but you can generate them via command line if you want. And then there's some other settings, which I'll talk about in a second. So I'm going to run, and it gives you an example of how to run just like a basic, very basic command. So I'll use that structure here. Just copy this directory. So I'm going to use a yeast database, even though these data files are mixed yeast and human, because it's just faster that way. But you can run multiple databases too. So I'll run cmd exe, and then the TOML flag. I'll put, I'll use the same TOML that we ran earlier. Then space and Spectra. I'll use multiple Spectra to show you how that works. And the reason I'm using quotes is because if the path name contains spaces, like task space settings, then Windows and Linux, et cetera, won't be able to interpret that correctly. So the quotes help it give it a literal path to follow. So the space, you can use spaces if you want in there. Okay. So then I'll either do for using multiple Spectra files. So you put -s space Spectra file and then just space again, and then put the next Spectra file path. I believe you can also specify a folder. Yeah, or folders containing Spectra. And that's just because if you have like hundreds of files, it gets pretty long. So you can just specify a directory if you want. So I could just specify the LFQ example directory, and that would be okay. So then I've got the TOML, I've got the Spectra, and then I just need a database. So I'll specify my yeast database. The FASTA. I didn't get an XML. And then this will produce a lot of output. You can change the verbosity with the -v flag. I'll run that again in a second. But you can just see that it runs properly. Oh, it'll ask you to confirm that you accept Thermo's license if you're running a thermo.raw file. So you just put yes. It'll save that for the future. You don't have to keep accepting it. And then it's doing the search. And I'm on a laptop, so it's a little bit slow. I might cancel it partway through to show you the other stuff.
Right, and so, I mean, you always got to think like, who's the audience? But if I, for some reason, I think about Jetstream because they have Linux VMs for free, right? Or free, sorry, subsidized is the word they want you to use. But let's say I wanted to use somebody else's resources, to run 100 files. This is the way I would use Metamorpheus in a Linux environment, right? Just to like crank through stuff.
It's close. I'll show you a couple of options for Linux in a second.
Okay.
So you can see it's doing the search. I'm just going to cancel it. Show you some other stuff. So this is the command I ran last time. So for Linux and Mac, running a Windows.exe will not work. So what you have to do instead is run .net. That's the command that runs. It's a Microsoft command. It's a Microsoft.net command. And then you specify Metamorpheus as an argument. So here it's cmd.dll, not exe. And so if you look here in the file structure, file directory, there's a cmd.dll. That's just .NET Core compatible DLL that if you pass it into the .NET command, it'll execute Metamorpheus. So it's the exact same thing For Windows, you can use either one, but for Linux and Mac, you have to use the .NET command and then specify the DLL. So I'll cancel that. You can also use some other flags. So verbosity will reduce the amount of output. --test. So that'll run like a very quick search. It's a very small slice of a yeast file, and it's large enough where you can run calibration on it. But it's very, very small so that it'll finish in a couple of seconds. And I'll specify an output directory. Oops. We call this the micro vignette.
I'm a big fan of the vignettes because, again, back to people that think that everything breaks. It's if you have something you're like, no, this works, I promise.
Yeah. Yeah, it's a quick sanity check to make sure that, because it could be any number of things, right? It could be your MZML file as it is something weird with that. There could be your database is weird. Like Metamorpheus is only one piece of the chain. So just to make sure Metamorpheus is working, yeah, the micro vignette's a good way to test that. I'll do minimal verbosity, for example. It'll tell you when each task is finished. So this is a calibration Gptmd search, and now it's done. So yeah, that's the micro vignette. You can also do those, of course, on Mac and Linux. Yeah, I think that's pretty much the command line stuff.
Did you want to mention the Docker, Rob?
Oh yeah, thanks. So another option, if you want to run Metamorpheus on, for example, a server, you can use Docker. I don't have Docker installed on this laptop, unfortunately, but you can pull a Docker image. And what that is it's basically it's an image with an operating system, .NET Core, and Metamorpheus all packaged in one. So you can actually just run it without any kind of prerequisites at all except Docker. So if you install Docker for Linux or Mac or whatever, you can run Metamorpheus without having to install anything. And that's useful for running Metamorpheus on servers. So latest is always the latest release. And then there's also specific versions. There's a dev tag, which is like kind of a debug version that you want to use, like the absolute, like if we push a hotfix, for example, but don't make a release, that'll be on the dev tag.
And you guys are updating these Docker images, right?
Yeah, it's actually automatic. Is that when a pull request gets merged in or a new release is made, then that gets pushed to Docker. So it actually happens all automatically without our intervention, which is good because humans make a lot of mistakes that computers don't make. So as long as you set it up correctly, it's a more consistent package deployment thing compared to having a human do it manually.
We like to try to incorporate as much automatic testing and automation into our workflow as possible. Because as Rob says, human makes mistakes. Computers, if you tell it what to do right, it's better off most of the time.
It also frees us up to not be fixing bugs constantly. We can focus on implementing new features that we want to make. And so it's just a much more efficient system this way.
Yeah, I mean, maybe I just haven't looked under the hood of most other places, but I find this very impressive. So also you're answering the question I wasn't going to get to, which was the whole bug testing. So appreciate that.
Yeah, there's a lot to that. I think probably at least 50% of our time is fixing bugs, but maybe it's actually a little high because we do have all these things that check for bugs.
I think it's like decreased over time until like we add a big new feature and then it like
break everything.
Yeah,
that's awesome.
And the last thing I want to show you is just MS Convert, which is not our software, but that will let you convert file formats to MZML. Like if you have, for example, a SciEx instrument or something like that, you can convert that to MZML and then run that with Metamorpheus. So me just Google MS-Convert really quick to show you. So it's a ProteaWizard tool. You can download it through SourceForge, which is kind of like GitHub. I've already downloaded it and installed it, so I'll just run. So I've run MS-Convert here, and then you can just browse for a Spectra file. So I'll find one of these raw files. So it's in thermo raw file format. You click Add. So it's in the queue to change. So the only thing you really have to do to get a Metamorphis compatible MZML is to centroid it. So profile mode raw files are fine because we use Thermo's software to centroid it, but we don't have our own centroiding software for MZML. So but MS-Convert does. So we rely on MS-Convert to centroid stuff for us before you put it into MetaMorpheus. So the way you do that, they call it peak picking. That's just centroiding. And then you just use the vendor one usually. And then they also say it must be the first filter. I'm not really sure if that's actually true, but I always put it on the first filter anyway. And that's pretty much it. You just hit start, and that converts the file for you. And then that will appear, I think, in this directory when it's done. But that's all you really have to do, and you can run that MZML through Metamorpheus. So you can see it's right here. It's writing currently. I think that's all I had to talk about.
Yeah, I mean, it's and just to confirm, I mean, because you guys, I mean, on the GitHub that you showed, you've got tutorials, you've got settings. So I'm sure, you know, like if you've got your timsTOF or iMobility, you're gonna have different settings, or, you know, blah 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 blah. You guys have a ton of documentation. So and Michael, you said you would put up like links to things that we discussed. Okay, cool. So no one had to like write it down, and there's not going to be things across the screen, except for they're like this 10 times he said Flash Gordon. I expect Flash Gordon to come across.
Yeah. I don't know if we have an animator on the team. Maybe Metamorpheus like needs its own like sort of speedy little superhero. But yes. I'm afraid what'll happen though is like the Michigan guys will make one that's like faster. So like, nothing good can come out of that. I think we don't want to start that war.
That's awesome.
But I mean, you mentioned the documentation that we have, and a great thing, if you don't find something there and you need help, or if we aren't doing something that you're interested in, just as Dr. Shorey mentioned before, just to reach out, like tweet us, issue on GitHub, email us. Like, we are very responsive and like to help our users within reason to achieve whatever they want. So
well, yeah, I mean, I guess we can wrap it up. So I mean, thank you for participating. Again, I think hopefully this is going to be fun. I know that if I was a student that had no idea, I would now be at least able to like click through it with you and believe that it works because you did it. Yep. And yeah, and like you said, hopefully we can do it again. I know there's so many other things, top-down, cross-linking, spritz.
Yeah, stuff. One thing I'm excited, of course we're excited to tell you about, you know, the other stuff that we're doing, but honestly, you know, because the community is so great, what I want to see is like, I want to see like videos from like other teams that are like working in this area. I want, you know, like, you know, other people to have a, you know, chance to showcase their stuff. And I think this is just great community building. Like you said, you know, we get a chance to know each other by having these conversations, and